Tissue culture techniques and their practical applications. Tissue culture is a term used for the process of growing cells artificially in the laboratory. Plant tissue culture is a technique of growing plant cells, tissues, and organs in an artificially prepared nutrient medium under aseptic condition. Techniques. The general technique of plant cell, tissue, and organ culture is almost the same with little variation for different plant materials. There are certain basic steps for the regeneration of a complete plant from an ex-plant cultured on nutrient medium. Tissue culture techniques involves the following steps. The first one is the selection of a suitable plant. Second is selection of a suitable part of the plant. Third is culture of the tissue. And fourth is propagation of plant and transportation. Coming to the first one, that is selection of the plant. This is the first step. Here, a plant which is economically important, that is having a medicinal value, food value, but fail to produce seeds or seed setting is low, or the life cycle is very long, that as in case of bamboo, can be selected for tissue culture micropropagation. Even tree species like teak, sandalwood may also be selected. The second step is the selection of a suitable part of the plant to be propagated. This may be a piece of tissue, or it can be a stem or axillary bud, or it can be even anther, embryo, seed, or even pollen grain. The third step is culture of the tissue. The tissue selected for the purpose must be separated from the plant, and this tissue is known as the ex-plant. This ex-plant must be sterilized. It must be washed thoroughly to remove the contaminants and placed in a culture medium. A suitable culture medium is prepared with special attention towards the objective of culture and the type of ex-plant to be cultured. Prepared culture media is transferred into sterilized vessels and then sterilized in autoclave. The sterilized ex-plant is inoculated, that is transferred on to the culture medium under aseptic conditions. Cultures are then incubated in the culture room where appropriate conditions of light, temperature, and humidity are provided for successful culturing. The cultured cells are then transferred to a fresh nutrient medium to obtain plantlets. The fourth step is propagation of plant and transportation. The plantlets growing in the tissue culture tubes or flask are very delicate and can be transferred to tiny pots and kept in protected greenhouses. This process is known as hardening. After the plantlets are several weeks old in greenhouse, they can be transplanted to the normal pots or to the field for further growth. The practical application of tissue culture technique. First one is germplasm conservation, mainly in the form of cryopreservation of some somatic embryos or shoot apices. Second application is technique of micropropagation for enhancing the rate of multiplication of economically important plants. Third is eradication of systemic disease in plants and raising disease-free plants. The practical application of tissue culture. Some clonal variations are useful sources of introduction of valuable genetic variations in plants. Second is it helps plants in imparting resistance to antibiotics, drought, salinity, diseases, etc. Next, the somatic hybrids and hybrids overcome species barriers and sexually incompatibility and produce hybrid plants with desired combination of traits. Next is the embryo culture helps in overcoming seed sterility and dormancy. Another application is the haploid production in culture helps to solve various problems of genetic studies and thus aid the plant breeders for producing new varieties. Another application is the production of synthetic seeds via somatic embryos. Differentiation for commercially important plants helps to achieve increased agricultural production. Plant tissue culture aid in producing the genetically transformed plants. The early flowering can be induced by in vitro culturing of plants so as to attain commercial benefits. The triploids as well as polyploid plants can also be produced by tissue culture technique for uses in plant breeding, horticulture and forestry. Seedless fruits and vegetables can be produced by following the endosperm culture method, which add to their commercial values. Next is increased nitrogen fixation ability can be achieved through association of tissue culture technique with genetic engineering. The callus cultures are useful in plant pathology as they act as an effective tool in the study of mechanism of disease resistance and susceptibility. Different tissue culture techniques helps us to study various biosynthetic processes, physiological changes and cytogenetic changes. Thank you.